Welcome to video 3 in our course G08 on metagenomics, particularly applied to viruses. So here we're going to look at metagenomic classification, taxonomic classification approaches, mainly focusing on how we're going to do it in a lab with Kraken 2. So our steps for the lab, we'll look at that. We'll do QC, which most of you have done before with FastQC, FastP, FastQ quality determiner, and multi-QC. And we'll focus on reassignment with Kraken 2 and then using their associated Python scripts to extract subsets of reads that based on the taxonomic ID before doing assembly and using checkv to get some summary reports on it. That's one approach. Another approach without Kraken 2 is simply to de novo assemble the whole metagenome using spades. We can check matches using log search. And again, check the context of interest using checkv. We can compare these approaches, and that'll be our kind of strategy for the practical aspect. So Kraken 2, we want to take our reads, assemble and find out more about them. And Kraken 2 can take those reads and identify at different taxonomic levels what they are most similar to. So it tries to classify them at the species, genus, family, and so on, different taxonomic levels. So this is very good for we're just trying to see what's there without having a good knowledge of this, and that's why Kraken 2 can be useful. And we have to consider our broader approaches, and that we may have typically metagenomes talk about shotgun sequencing, where we don't really have the option of marker genes. We're looking at processing the data, and later we can do functional database searches to find out more information. So that's why the absence of marker genes means that this taxonomy assignment is more tricky, and therefore we can use things like Kraken 2, which is it's just one of many tools. Amplicon-based sequencing can use OTUs, so that's outlier taxonomic units, or ASAPs, which are basically SNPs, and that can also be used in this process as well. So OTUs in bacteria tend to operate at the genus level based on certain genes that are of interest, such as the 16S. Typically, you can identify their identity up to about 97% gives a distinctiveness. So if their identity is greater than 97%, they are allocated to the same OTU, which is equivalent to a genus. And this is effective for bacteria, and that's based on the DNA sequence alignment, where you can find ones that are a bit divergent and therefore can contain more differences. So you can see here, these three here have some nucleotide difference compared to the ones before, like at this A site here, and there's other examples as well elsewhere, like the C's here. So this works well for bacteria, but less well for looking at viruses. However, you can use viral operation taxonomic units if you've substantial coverage of the whole metagenomes. Dereplication can be relevant for that if you have a lot of different assembled genomes, and therefore we can apply standard metagenomic metrics like alpha base diversity and beta diversity to that. Alpha diversity refers to the within OTU diversity, whereas beta is across OTU diversity in simple terms. What we're also going to think about is metagenome binning. So what Kraken 2 will help us do is allocate contigs or reads to different species to make up metagenome associated genomes. So often our understanding of which contig belongs to which species is unclear. So this is in the Kraken 2 approach that you can see here, where we have our reads, we assemble them into contigs, and we need to allocate them into bins to differentiate them from each other, which is not always straightforward. There's lots and lots of ones that do this. These ones are about old, but you can see there's lots and lots of options. So this just gives you a sense that there's many ways of approaching these different strategies, and we're just using one approach, really, well, two shared approaches, one of which is dependent on on Kraken-2. Another thing we can do is use BLAST or other searching approaches to assess our context, and that can tell us more about to which organism it belongs. So Kraken-2 is useful that it looks at not just viral reads, but all the reads that are present. So if we have hosts or other contaminants inside there, it should be able to allocate them to the appropriate representative right across the tree of life. Another one that will be mentioned later is DIB-FREE Finder, which is a reference-free and alignment-free approach that tries to do viral content assignment as well. So there's lots and lots of metagenomic databases, and there's very, very many. Kraken-2 is just one of them. The ENA has over 50,000 metagenomic assemblies, most of which come from Magnify. IMGM has over 30,000 of them. The NCBI Genome Assembly Database has 25 terabytes of sequences, and that's why you often have to use the online version to search, because it's not really logical to download all of this info. And the SRA has over 23 million accessions, so that kind of gives you a sense of the diverse databases against which you could compare your metagenome of interest. There's also the IMGVR, which is over 5 million viral genomes. Often these are environmental and host-associated ones. They're contain both isolated viruses and also metagenome assembled ones. And this database, we're not going to use in the labs, but it does have options to perform taxonomic filtering, genome similarity, and gene content analysis. Another one is the NCBI RefSeq viral database, which is 10,000 viral genomes from cultured and studied viruses. They're typically high quality ones, and this is good for viral annotation and classification, but it does have a limited environmental diversity. Another option is Viper, the virus pathogen resource. Focuses more on pathogenic viruses, and has genome sequences, protein structures, host immune interactions, and more specialized data sets such as flu, dengue, Ebola, SARS-CoV-2, and so on. Allows you to do more nuanced analysis such as multi-sequence alignment, 3D visualization, and phylogenetic analysis too. We're going to focus on Kraken-2, and advantageously we have the databases already locally on ORACs, which makes it more easy if you're trying to create and these databases on their own. It's not always straightforward, and you need considerable space as well. And Kraken-2 is also a high RAM tool, so you need sufficient RAM to make it run effectively. One key thing of interest is that it takes the reads, and for given a small set of k-mers, it will use a minimizer as a representative subsequence, where L is some length less than the length k, and that minimizer will be the query that is assessed. And it tries to allocate this to the most general taxonomic category possible. So if you can allocate it to genus instead of species, it will do that. Like vice versa, you can do it to family. If it's present in all the members of the family, it will allocate to the family level, and so on. So it's the lowest taxonomic, lowest common ancestor is allocated the read where possible. And it uses minimizers as in effect it's subsampling from the reads, the read parts, the sequences on the read, and therefore it has a small error rate associated with that process to make it more efficient, given the number of comparisons that are going. It can also handle amino acid sequence as well as DNA sequence. So we can imagine that we have some k-mer from a read here, and what we'll do is actually take some length, like the pink length L34 there, and use this as our search query, and we'll create a hash from this and use a k-mer lookup process to make a table from this. So we'll record the matches in a compact hash table and identify the LCA, the lowest common ancestor associated with that match, and link those together. So in our output from Kraken 2, we'll basically have the taxonomic ID at the whatever level, species, genus, family, order, and so on that's appropriate for it, and that's directly linked to the minimizers representing the sequence as well. And this is a visualization of the LCA in case you're unclear. That if it's present in all the members of a species, then we can allocate it to the genus. If it's present in all the members of the genera inside a family, it'll like allocate it to the family, and so on and so on and so on and so on. The output looks like this. So we have information here as to whether it's classified or unclassified, U and C. Some of the lines are bigger and shorter, hence it's a bit different lengths here. You have the header of the sequence read that's being explored. You see for the classified ones, we have the taxonomic IDs here. It's not the most clear necessarily in this format, but that's the taxonomic ID that's in that column. And what you can see then, we've information on the matches to the different taxonomic IDs. So you can see there it has a taxonomic ID and the number of hits associated with that. So here we have 5 hits, and on the right we have another taxonomic ID and the number of hits. So this information is more visible here, where we have the fraction of the total reads over here, and this is the taxonomic ID here, the taxonomic code. So you can see that we have a lot of some rhodovector, but as we go up, we see increasing allocation of reads at different levels. So many organisms will be represented in these kind of tables, but at low levels. So if you have something with fewer than 100 reads, it's unlikely to be, it's possibly a contaminant that accidentally happened during the sample prep or sequencing. It's unlikely that it is something that is reproducible. So I know there's other ones that are commonly used as well. We're just using Kraken 2, for example. Kaiju is another one that can be very effective. It compare reads to a reference protein database of microbial and viral protein sequences. The non-redundant RefSeq database is an example of that. It uses Burroughs Reader's transform to find the minimum exact matches, allowing for some mismatches. And it also does some DNA sequence comparisons to check that the match is accurate. But we need to make the databases for that, just like Kraken 2, which we don't have. So we're not going to use this particular one here. So that kind of concludes our part here on metagenomic classification with Kraken 2.